So we will quickly look into how real-time PCR-based gene expression can be conducted. What are some of the theoretical aspects, practical aspects, and the troubleshooting, right? If we look into the basics of the real-time PCR, there are two major division you can make. One is the wet lab experiment, uh, and the second one is the software or the computer-based work. Within the wet lab work, if you see, the first step is to collect the sample. Second is that the preservation of the sample has to be done in a proper way, followed by RNA extraction. And what are the different things when we have to do the RNA extraction that we will see. Once the RNA is extracted, you have to convert it into cDNA, but before that, you have to treat it with the DNase enzyme. After that, you have to look into the quality and the quantity of the RNA, and then a particular volume or the amount of the RNA you have to take for the cDNA preparation. Once you make the cDNA preparation, how you perform the real-time PCR, that we will look into it in a brief. Once you do the real-time PCR in the thermal cycler, you will get the data, and then we will go with the analysis part, from where the computer-based or the dry lab starts. In order to do the data analysis, we have to look into several different points, like we have to check for the primers, we have to check for the reference gene, we have to check for the relative expression. So in primer, like primer designing, what is the primer efficiency, specificity, that we will look. In the reference gene, different algorithms which are used to find out the reference gene, that we will see. And then finally, once everything is done, we will go for the relative expression. And as you can see that there are several different methods which are generally used for the relative expression. For example, delta CT method or buffer method, right? So we will look into all of those in one by one in different videos. We will go into the basic first about the wet lab, or how we, what are the steps involved in the wet lab. But before that, it is very much important to understand that what gene expression is. Gene expression, you can consider it in a way that it is a like a on-off switch or a volume control switch. So whenever any condition is encountered by the cell, some certain genes, it needs to be switched on or switch off. And that happens, if you remember well, the central dogma of the molecular biology, there are genes which is being transcripted into RNA, that is mostly messenger RNA or mRNA, that further involves in the translation. It means the formation of the amino acid and then peptides, which ultimately makes the different proteins. Many of them are enzymes, and then finally the cell uses those enzymes to counteract on the environment what it has encountered. So gene expression is that part where the gene is being transcribed into the mRNA. So if the situation arrives and the gene needs to be turned on or off, that is where the gene expression comes into play. And sometimes the same gene, even though if it is expressed in certain quantity or the copy number within the cell, that needs to be up or the down regulated given the different circumstances. So basically, it's a on-off switch or the volume control switch to determine how much of the gene product has to be made by the cell, right? Gene expression change is one of the major component. If you look into the acute stress response, which actively immediately reprogram the metabolism, cell cycle progression, protein homeostasis, cytoskeleton organization, and many of the enzymatic activities modulation. If you want to look more closely that what the gene expression in response to stress, or the gene expression in response to environmental condition, how it contributes to the rapid response to the evolutionary adaptation, I would suggest you to read these two review papers which has been published in the Nature, right? So these gene expression could be specific to a particular stress, or it can be combination of different it could be common for different stresses. For example, when we talk about the oxidative stress, it's a very common response by the cell. Uh, which can happen even in the cold stress, heat stress, encountering with the pathogens or other biotic or the abiotic stress responses. Now, these gene expression modulation could provide acute stress response, or it could lead to the acclimatory or the adaptive response in the cell, and that could be involved in the adaptation or the acclimation of the organism under the different environmental conditions. When you talk about the gene expression, it can be studied either at an individual gene level. For example, let's say if somebody is studying that how the climate change or how the temperature increase is relevant or affecting, let's say, shrimp, for example, then we may focus only on HSP70 gene. So when you study those kind of individual gene or pathway related gene, or when you know that only when your research interest is only for some particular pathway and selected important enzymes, then you are not concerned about what is happening to other pathways and other genes within the cell. So in that case, you go with the individual gene level gene expression study. Or if you would like to see that how overall, because of, let's say, a decreased pH, the cell has responded, then in that case, you go with the global scale studies, in particular, for example, RNA sequencing or the transcriptomic analysis, right? So over the year, the research in the field has progressed from candidate gene expression to global approach. Basically, candidate gene expression in the very beginning was studied using a process called as northern blotting. As many of you are already aware that when we talk about these blotting, it means that your biomolecule is being transferred on the filter paper or any membrane, and then that is being hybridized with a particular probe, and using this probe and on the filter or on the membrane paper, you use the X-ray film to see that how much expression has happened, right? Similar to how we do Western blot for protein or Southern blot for DNA. From there, with the advent of the PCR, the real-time PCR is also becoming very common for the gene expression studies. Other methods to do it, like express sequence tags or SAGE analysis, that is serial analysis of gene expression. And with the advent of the next generation sequencing, it has become much more and more common to do the RNA-seq or the global stress response for the gene expression studies in any of the organism, right? So I'll not go in detail, but you can see that there is a northern blot, there is a wherein, as I said, that the gene or the transcript is being transferred on the membrane, and that membrane is being labeled with the probes, and after the probe labeling, you can develop it into the X-ray film and check that how much gene expression has happened. Real-time qPCR, that anyway we'll look in more detail in the coming videos and the slides. You also have methods like express sequence tag, where you sequence the cDNA library and then check that how many copy number or how many times that particular sequence is or the gene or the transcript is present in your sample. Similarly, we have the SAGE analysis. I'm not going in detail because nowadays nobody is doing SAGE, or for that matter, even microarray. Microarray was once considered as a very powerful tool, but wherein you label the cDNA on a very small microchip or microarray chip, and then you hybridize it with the probe, and depending on how much is the fluorescence or how much is the hybridization, you get the values in the form of fluorescence, and then that detects and tells you that what is the gene expression. However, there were several drawbacks with the microarray hybridization. Basically, you have to know the genome or the transcriptome prior for the organism. You have to custom made the microarray in order to check the hybridization, and many a times that you cannot do it for the entire gene or the transcripts present in the cell. That's why nowadays even microarray has become a very obsolete technology. So basically, real-time PCR and the RNA sequencing are the two most common ways in which we study the gene expression. And in RNA-seq, as I already said, that you extract the RNA, you convert it to cDNA, you do the fragmentation, you prepare the library, and then you look at what is the copy number of each and every exons or each and every transcripts in the whole transcriptome, and then based on that you determine that whether which is upregulated or which is downregulated. So as I said, that there are several methods, and each are having their its pros and each are having their its cons. You can go in more detail if you are if you are interested in understanding the different technologies or different methods used for the gene expression studies. I have given you the reference for the Illumina techniques, right? If you are interested, go for a better understanding pros cons of different methods, what I mean, when in what circumstances you chose which of the method of the gene expression for your studies, right? So coming back now, we know that what gene expression are and what are the different ways or what are the different techniques which is used for the gene expression studies, and a bit of basic. Now we come back to the laboratory method. As I said, the first and the very important step is the sample collection or post-experiment storage of the sample, right? So, or the preservation of the sample. Different sample bring different complexity when you talk about the RNA extraction. Example, in the polyphenols, you have the polyphenols in the plants and the algae are very much difficult to remove when you do the RNA extraction. Similarly, there are the cases where you have much higher DNA or much higher secondary metabolites that again cause difficulty in order to get a high quality RNA, right? So we will look at how we solve those things overall in different samples. But the first and the foremost important step is to preserve the sample either after the collection from the field or post experiment. Time and again, people have looked into different methods more suitable for the RNA storage for gene expression studies. Some of the papers, for example, one which I am citing here, has suggested that the tissues, in particular for the freshwater insect in the remote habitat, can be stored in molecular grade ethanol. If you are more interested that how this ethanol has worked for the RNA preservation, I would suggest you to read this paper. Second important thing, which even I was following when I was doing work with the algae, is a flash freezing the samples in the liquid nitrogen and then store directly in -80. However, the drawback with this method is that sometimes you are not having the possibility to get the liquid nitrogen in the field of the site where you are collecting the sample, right? If it is in the laboratory condition, well, you will have the liquid nitrogen and you can do the flash froze and then keep immediately into the -80. However, in case of field, you may not have the accessibility for both of them, that is the liquid nitrogen and -80. And if you are choosing this method, then it is advisable that you make an aliquot of the sample before preserving in -80, because every time you take the tissue out from the -80 in order to collect the small aliquot of the sample for the RNA extraction, there is a degradation of the RNA which is there in the sample, right? Every time you do the fridge and thaw, almost 20 to 30% of the RNA concentration you will lose every time you do it, and close to 3 to 4 months you can store the samples in -80 if you are going with this method, right? Many people also does that. They homogenize the tissue immediately after the experiment or after the harvesting of the sample in a chaotropic based cell lysis solution, for example, intrizol, right? So in trizol, you have a lot of guanidinium, which helps the RNAs to inhibit, and then in that way, you maintain your RNA concentration in the sample well, right? The most famous, or you can see that the most used way to preserve the sample is RNA latter. RNA latter is again uh, a very high salt containing preservative. Many different alternative commercial names are there for RNA latter. Different company has got patent for different names, and there are also recipes which are available online. If you go to buy RNA latter, it is sometimes maybe very expensive. You can also make it in lab and it works very well. So I have given you the reference here to learn more about the RNA ladder from the Thermo Fisher website. What is important when you collect or when you preserve the tissue in RNA ladder is that the sample should be smaller than 0.5 centimeter, and generally you should take at least 5 times of the tissue. For example, 0.5 gram of tissue needs at least 2.5 ml of RNA ladder to store, right? Another thing is that if you think the sample are hard to break or the RNA ladder is difficult to penetrate within the tissue, it is advisable that you cut it into small pieces or you make some holes in the tissue so that the RNA ladder enters into the tissue well, and then it preserves the RNA. Another important thing is that whenever you are doing the RNA ladder preservation, you cannot do it in the preserved sample. So you have to do it into the fresh sample so that RNA ladder enters within the cell, right, or within the tissues and protect the RNA. You have to generally keep a day post RNA ladder preservation at room temperature, followed by you can either keep, if you want to keep it for short term, let's say 1 week, then you can keep it in the fridge. If you would like to keep it for longer duration, then it is advisable after a day of incubation in the room temperature, you should bring it to -20 and then store it for a few weeks, or sorry, few months, right? I hope that now it is clear that the sample preservation is a very important step when you do the RNA extraction. Now you have preserved the sample. Now the time comes when you do the RNA extraction. There are different methods for RNA extraction. Sometime for some of the tissues, the kit method works really well. For some tissues, the manual method works really well. And in many times, you have to combine the kit and the manual method in order to get the high quality and the quantity of RNA, right? So for different tissues, you have the different RNA extraction, and even within the RNA, let's say if you're focusing only on the small RNA, or if you're focusing on some certain types of RNA, then the kit varies. So you have to look into different company websites, and then you have to decide that which RNA extraction method researchers across the globe have been using and are more successful, right? Example I have given you here, for example, CTAB, which works well for the RNA from the high recalcitrant plant tissues or the algae, while Trisol is more ideal for those samples which are high in DNAs or high in the lipid content, right? If you would like to know more about what sort of different RNA extraction methods are, there are very good resources by Promega, which the link is already given here. If you're interested, you can have a look into it. Whichever method of RNA extraction you chose for your sample, what is important is that when you perform the RNA extraction, you have to be very careful. Careful in meaning that the clean working area, right? Workbench, pipette, sterilized consumables, all are very much necessary. You have to use the lab coat or apron, or otherwise you don't have to, touch your clothes very much time. When you are using the gloves, you don't have to touch your skin, because even in the sweat or even in the body fluids, you have a lot of ribonucleases which generally cleaves the RNA, and then your RNA extraction becomes more difficult, right? So you have to be very fast when you are doing the RNA extraction procedure. You cannot keep the tissues or the cells without a buffer for a longer time, because then the lysis of the degradation of the RNA happens before you do the lysis, right? There should not be any group discussion, gossip talk, or any sort of collaborative work when you're doing the RNA extraction at a particular place. So you have to just work very quietly, very calmly, with full attention when you do the RNA extraction. Another thing which I generally suggest is that if you have to do the RNA extraction, you should do first thing in the morning, because at that time you are much more attentive, and the chances of getting anything wrong is much less than working in the evening or late evening when you are already exhausted with the full day of work, right? You have to use DEPC treated water for the reagent preparation, not necessarily, but if you have doubt that your distilled water or your milky water is not like giving you more trouble, then better to use DEPC treated water. Now DEPC also has how you will prepare it is by adding 0.05 or 0.1% DEPC mix in water. Mix it really well for at least for 15 to 20 minutes continuously, or otherwise mix it for 5 minutes, keep it on the working bench, mix it again, keep it, mix it, keep it, mix it, keep it. So at least do it for 30 to 40 minutes. Once you are very much confident that the water is fully mixed with the DEPC, then in that case you can keep it overnight for and then the next morning you take it out, you do the autoclave. It is very important to do the autoclave because then DEPC has to be degraded before you start or before you use for any other purpose, right? Some of the reagents, for example tris, may not be compatible with DEPC, so you always have to make such kind of buffer in DEPC treated water. It means after the autoclave, when you are very much sure that DEPC is not there in the water, right? And if your buffers or the reagent are compatible with DEPC, you can even make it before autoclaving and then you can autoclave, right? You have to thoroughly clean the gel running apparatus. Sometimes people also use NaOH, that is a bit of alkaline condition, alkaline water to clean your trough and the gel trays and comb for the RNA. And it is always advisable to make a separate TAE or TBE buffer, whichever you are using, for the gel loading dye, for the RNA purpose or RNA work. And you can use gel packs or the ice trays or the crushed ice to keep the reagents or keep the RNA in between the procedure, right? So basically, we have to protect the RNA from hard to inactivate RNAs. There are several commercial products like RNase Away or RNase Zap, which is widely used across the world in the laboratory to as a decontaminating solution. However, you can also make RNase chap or similar decontaminating solution into the laboratory, for which I have already given you a link. And I don't think it's very difficult to make it. But again, if you are going to buy it, it's going to be a bit expensive. I guess close to 17,000 to 20,000 rupees per liter. And you can make it very easily with not much expensive chemical in the laboratory, right? Whichever method you choose for RNA, irrespective of the method, it is most likely that the RNA extraction is having the DNA, because most of the RNA extraction method without DNA removal is incapable to fully remove DNA. And when you do the real-time PCR is not capable to distinguish between the cDNA, which is synthesized from your RNA by the reverse transcription, and the contaminating genomic DNA. So what will happen is that when you are doing the PCR, if the gDNA is also contributing to your template, then there will be an overestimation of the amount of the RNA present or the gene expression, right? So this could be a problematic result, or it could not be a very correct result when you do this kind of mistake. So generally you have to avoid that. And how would you avoid it? First way is also that you make the contrasting primers to span the intron boundaries. However, it is sometimes difficult to make, and why should we complicate things? So in order to avoid that, DNase 1 digestion is the most commonly sought method to remove or remove the DNA from the extracted RNA, right? And different companies gives different instructions to use their DNAs for the RNA extraction, right? So have a look into it. There are also possibility of gel fractionation, acid phenol chloroform extraction, and last but not the least, lithium chloride precipitation. However, my personal experience is that even with the lithium chloride precipitation, we often get DNA in the extracted RNA, right? If you are interested more to know about the DNA's treatment or the DNA contamination in the RNA, I have given you a couple of resources here, which you would find it interesting if you want to get more information about the DNA's treatment of the RNA, right? In particular, the third one, that is a book chapter in a published book called as Molecular Analysis in the Archive Tissues, Methods in Molecular Biology, I guess. In that, there is a paper by Isabella and Serena, and they have talked more about different DNS treatment of the RNA, right? So have a look into it if you are interested in understanding or getting more information, right? I will also give you the information on the DNase one which we use in the lab, right? So DNase one degrades the single strand or the double strand DNA, right? It is supplied at a concentration of 1 unit per microliter, and in general, this particular DNase works 1 unit per 1 microgram of RNA. So how would you know that? So suppose if you do the RNA extraction and you do the quantification. So how do we do the quantification? That will come in the next slide. But let us suppose that you have 400 nanogram per microliter concentration of RNA, which you are expecting to have a DNA as well, right? So you will need 2.5 microliter of this particular RNA, right? For example, in the C1. And then, for example, in C2, if the concentration is 350 and C3 concentration is 600, so you can see that 2.5 microliter of C1, 2.86 of C2, and 1.67 of C3 probably will have a RNA concentration of 1 microgram. And for that volume, you need to take 1 microliter of the DNase enzyme, because each microliter is having 1 unit, and 1 unit is sufficient for 1 microgram of RNA, right? You do this particular calculation, since the DNase buffer is most of the time 10x, as you can also see. Here, it's 10x, right? Here. So 10x will be required one if the final volume is 10. Remaining thing you have to make up with the nucleus free water, and then you will get 10 micro, right? So similarly for this C2, you need 2.86, 5.14, and for C3, 1.67. You will see that your buffer and the DNA remains same. Volume of RNA is being compensated by nucleus free water in order to make a final volume of 10 microliter. So that is how we use RNA free DNase in our laboratory, right? Once you do the DNase treatment, it is very important that even before going for the cDNA synthesis, you look into the quality and the quantity of RNA. Traditionally, what we do is that in order to understand the quantity first, we do the spectrophotometric quantification, right? The concept remains same, that absorbance at 260, which is common for the DNA and RNA, gives you the concentration of the RNA and DNA, right? Either you can do on spectrophotometer, you can do on nanodrop, or you can do on multimode plate reader. Everything remains same, right? I mean, concept, that is 260 absorbance gives you the concentration. Now, for 1 centimeter path length, when you are using, let's say, a spectrophotometer, 1 centimeter path length OD 260, if the value is 1, it means the absorbance at 260, if the value is 1, means 40 microgram per ml of RNA, right? If you keep that in mind, then RNA concentration will be 40 microgram per microliter multiplied by OD. So let's say if you are getting 0.25, right? So multiply here 0.25, and then whatever the dilution factor you have used in order to make use into the spectrophotometer. For example, let's say 1 microliter of the RNA, extracted RNA, you have diluted it to diluted in 199 microliter of water. So that is 200 times you have diluted. So your RNA concentration will be 40 into OD 260. Let's say if it is 0.25, then 40 into 0.25 multiplied by 200, right? And since we are using 1 centimeter path length, so everything will be divided by 1. If you are using different path length, then that you need to consider when you are calculating in the equation by putting here the path length. Okay, everything divided by path length, right? That you check carefully, right? So that's how you do. So if you look here at 260, you get the DNA and RNA, and 280 you get protein, and then you get salt. So even though RNA concentration, it is important to consider 260, and then you get the value, you get the quantification. But quality is equally important. If the protein is high, and if you see a second peak here, or if the salt is high, and if you see a second peak here, then that's a problem, because even though you have a good quantity of RNA, but then there is a protein as well as salt or any other exopolysaccharides, there could be any other salts in the extracted compound, which may interfere when you go with the downstream process of the cDNA synthesis or the qPCR, right? So how do we check it? OD at 260 by 280, right? OD at 260 by 280, for if it is a pure RNA, the value should be around 2. However, between 1.8 to 2.1 is considered a good value for the OD 260 by 280 for RNA. Sometimes people also do, researcher also do the reading at 320 and then make 260 by 280, 260 by, sorry, 230 by 320, and so and so on, right? So what happens is that those values are also somewhere should be around 2, so that you can say that your RNA is pure, right? This is a traditional method. Now, more and more fluorometric methods are coming, wherein you have the, for example, qubit fluorometer, which is dependent on the fluorescence. There is a there are reagents from the Invitrogen, for example, which is compatible with qubit fluorometer, wherein you add your RNA in that particular reagent or solution, and then you get the reading in qubit fluorometer. So I'm not going in detail about how qubit fluorometer works, but if you're interested, have a look into this thermofacial link which I have given here, right? The third one is the Agilent bioanalyzer, which is a very advanced way of doing the quality and the quantity check. You can see here it runs like a gel on a chip. So there are chip which is for, let's say, plant DNA, plant RNA, animal DNA, animal RNA, organelle DNA, organelle RNA, something like that. There are several different chips which comes, and you load your 1 microliter of the sample on that chip, right? Once you load 1 microliter of the sample in your chip, you always have a ladder in the kit, and then you will have distilled water as a negative control, and then you can see when you run it, it takes less than 2 minutes, 20 seconds to 80 seconds here, right? And then you will see that there is a separation of the ladder and then your sample. For example, here I am giving you one of the sample, my sample when I was doing it earlier, and then you can see that there are two distinct bands. One is here, uh, one is here, and the other one is here, right? In between, you have some more bands, very light thin bands, but those could be also because of the chloroplast and the mitochondrial DNA and the RNA or the nucleic acid, which is contributing to it. But majorly, you will get this two, right? And then this electrophenogram, if you look here, you have one band at 45 second, and then the other major band at 40 second, right? And then along with that there are few, and this is for the distilled water, right? So this will be there always in the water. For example, you can see here, right, the DPC water, right? This value or this level should be double than this level, then it is considered good quality or the high integrity of RNA, right? Lesser the peak, like two distinct peak, right? Like this is considered a very good quality and the quantity, quality of the RNA. And it also gives you values in terms of relative RNA integrative number, relative integrative number. And if it crosses 7, for example, here 7.3, 7.8, it should be sufficient to conduct the RNA sequencing or different downstream processes, right? right? Uh, Most often, we will not do Agilent bioanalyzer or qubit. We will be doing the spectrophotometric reading and then running the gel. When you run the gel, it is more or less similar to DNA. However, RNA may have the secondary structures, and that may cause like not a uniform run in the gel. As a result of which RNA we generally run on the denatured agarose gel. However, we and most of the lab are using similar agarose gel conditions how we do in the RNA. Uh, what is important here is to check the ratio of the 28S and the 18S band. Why 28 and 18S? Because we know that tRNA tRNA, rRNA, and mRNA. There are three different types of RNA, right? rRNA contributes to close to 80 to 85% of the RNA present in the cell. So when you are doing the total RNA extraction, 80 to 85% of your RNA is coming from rRNA. And rRNA, let's say you have, though you have several different subunits, 18S and 28S in eukaryotic cell are the most uh, uh, abundant ones, right? So 18S and 28S. So if the RNA integrity is good, or if the extracted RNA is of good quality, you will always see that 28S band should be double than 18S band, right? Um, if it is not so, then you can say that the RNA is degraded while doing the extraction. If it is even equal or anything like the down, let's say 18S is thicker than 28S, then the RNA is degraded, right? If it is for one or two individual gene, it should not, or it may not impact much your gene expression. But if it is for RNA-seq, then that will cause a problem in the downstream processing, right? So important. I have also given here the working with RNA, general analysis of RNA, wherein you can see that there is a formaldehyde gel, right, which is being used as a denatured gel. However, majority of the time we will just do the agarose gel, and then not in the MOPS buffer. We are doing in the TAE buffer, which should be fine, right? And this is just for your understanding and learning. If you want to if you want to know more, like how you can look into the quality and the quantity of RNA, I would suggest you to go into the Thermo Fisher website, and then you can get more information about it. Once you do the DNA treatment, right, what I suggest is that you should immediately convert the RNA into cDNA, because if you are preserving your RNA in -20, then the stability is very less, and then the chances of getting it degraded will be much higher. Even if you're putting it into the -80, every fridge thaw will cause a lot of degradation in the RNA. If you are taking it out after a month, or if you're taking it out frequently to use for the cDNA conversion, right? Another thing is that if you're storing the RNA in -80 or -20, then you should do every time quantification before to convert into cDNA, because as I said that I mean, storing the RNA in -20 or -80 cause some degradation. So let's say if today you are doing it and then finding concentration of 600 nanogram per microliter, and then based on that, if you do the cDNA conversion of that particular sample after a month, it may not be correct. So after a month, when you are taking out the RNA again from the -80, then at that time you have to do the quantification again, and then based on that you have to make the cDNA conversion, right? So I hope that's clear now. You got some idea. Uh, Let us go into the next one. Once you have extracted the RNA, treated it with the DNA, DNAs, you check the quality, you check the quantity. Now it's time to do the conversion into the cDNA. How will you do it? This is the first step where you need to normalize the RNA level from the different sample, because if you have to do the relative gene expression, it means if you have to compare the sample 1, sample 2, and sample 3 of the control and the experiment, let's say E1, E2, and E3, if you are not normalizing the initial RNA, then the chances of getting biased expression will be much higher. it is in the same way, like let's say if I am saying that uh, the sample one is having I mean with the method of the RNA extraction and all the process which you did, you get 400 nanogram per microliter, and in the second sample you got 350, third sample you got 600. So if you will use uniformly 2 microliter from each three of the sample, then what will happen? For C1 you took 800 nanogram final volume of RNA. For C2, you took 700, and for C3, you took 1.2 microgram or 1200 nanogram, right? So initial material, if it is different, then you will not be able to compare the gene expression. So for that, you have to normalize it and then take the equal volume of RNA from each of the sample. So for example, here for C1, if the concentration is 400 nanogram per microliter, then for 1 microgram RNA, you will take 2.5 microliter, right? Similarly, for C2, if the concentration is 350, then you should take 2.86 microgram of RNA. And for C3, 600 nanogram per microliter, so you should take 1.67 microliter of RNA, right? That variable, that volume of the RNA is variable for each and every sample. And again, as I said, even before, when you were doing the DNA treatment, whatever the difference is coming in the RNA volume has to be normalized or has to be complemented with the nucleus-free water. That we will anyway see in the next slide, right? So different cDNA synthesis kit is having somewhat different reaction mixture and somewhat different thermal cycle profile. So whichever company and whichever model you are working or you are having in the lab, that manual or the practical guide has to be downloaded from the internet, or if they have supplied along with the equipment, I mean reagent, that you have to look into it, right? For example, iScript Biorad, let's say here I have given you the component and the volume per reaction, and in the next slide, if what we are using in our lab is Promega. So I have also given you some idea about Promega, what we have, that is the Go script reverse transcriptase. The volume of the RNA again depends on, as I said in the previous slide, will vary for each of the sample. Here in the iScript, if you remember, you were using one single reaction. Whereas in this, you do in two different steps. In the first step, you prepare your component one, that is where your RNA is with some random primer, nucleus-free water. You heat it for 5 minutes at 70 degree. And then from there, you take the second step, right? And the second step or the second component is having your different buffer, your coenzymes, cofactors, so and so on, nucleus-free water for 15 microliter. And then only after first and two component you mix, you go for the thermal profile, which is here, let's say 25 degree for 5 minutes. I don't know if you can see here, but I'll show you 25 degree for 5 minutes and then 42 degree for 1 hour, right? And then how you can look into the process, that how you stop the reaction, right? So that is how that is what I just wanted to explain in the cDNA conversion. When you do the cDNA preparation, right, you have to check the quality of the cDNA. How would you do it? The best way is to take the cDNA, dilute it to 1:10 times. So now 20 microliter of the final volume of cDNA became 200 microliter. Now from that 200 microliter, take 1 microliter as a template and either run a normal or a regular PCR, conventional PCR, or real-time PCR using, if it is a real-time PCR, use the cybergreen. If it is a conventional PCR, use the tag DNA polymerase with a known thermal profile for that particular gene which you have standardized in the lab, right? Check if the amplification is happening or not. And anyway, you will see that if the amplification has happened or not. In the regular gel, you will see using the DNA ladder, and if the product size is expected to what you are expecting or not. If it is a real-time PCR, you will be able to see the amplification, and then you will be able to see the relative fluorescence unit, which is coming in the y-axis, as well as using the belt curve analysis, you will be able to see whether it's a dimer or whether it's a actual product. That anyway you will see in the coming videos and slides, right? So that is very much important. 2 points to remember. One is that cDNA has to be diluted before to perform the qPCR, and second one is that before to perform the qPCR, you have to test that whether the cDNA which you have synthesized is correct or not, right? Now we look the sample preparation, we look the sample preservation, RNA extraction, DNA treatment, quality, quantity, how do we do it, and then finally the cDNA conversion. Once the cDNA is converted, you have to set up the qPCR. Again, the reaction mixture recipe depend on the different manufacturers, right? And what sort of cybergreen you are using, whether you are using fast cybergreen, whether you're using standard cybergreen, whether the cybergreen is coming from Biorad or Promega or Thermo or whatever the company you are using, right? For each and every company, they will give you the protocol, and basically you should follow that, right? For example, Promega we use in the lab, and I have shared here, right? So you can see that the qPCR master mix or the cybergreen master mix is 2x given. So you have to convert into 10 microliter. So if the final volume of the reaction is 20 microliter, you have to take 10 microliter of the cybergreen. You have to take a primer forward and reverse in the range of 200 nanometer to 1 micro, not micrometer, 200 nanomolar to 1 micromolar. All right. Basically, we take is 10 micromolar concentration, 10 micromolar concentration, and then from there 1 microliter. So in a 20 microliter reaction, it becomes how much? 20 times less of 10 micromolar. So basically it becomes 0.5 micromolar, which is, as you can see, in the range of the final concentration, which is suggested 200 nanomolar to 1 micromolar, right? So it becomes 500 nanomolar if I see in that way, right? For forward and reverse both. And then you don't have to add the CXR reference dye, because generally there is already a control reference dye, right? In the thermal cycler these days, right? There is already a control. You don't have to add that. And then you have to make up the final volume reaction to 20 microliter. Once you do the reaction, it is very important that the tube in which you have done the reaction has to be centrifuged, because we do not want a situation wherein one or the other component stays on the wall and does not mix well with the reaction mixture. When you do the centrifuge, make sure that you do not do it at a very high RPM. You should do it, let's say 3000 or 4000 RPM at the max for 1 minute or 2 minutes, right? But do not mix it at a very high RPM, because then your enzyme will get degrade, or some of the, there could be some compromise in the reaction, right? So do that. If you're doing on the tube, label it properly, starting from A to H, and then you have to make a similar drawing in the notebook to know that in which lane, which sample it is. Because sometimes with the real time, when you're doing with more number of sample, getting confused is very much common in filling those 96 wells, right? Or the strips. So that you have to take care. And after that, there is a thermal cycling condition. It depends on whether you are using fast cybergreen or standard cybergreen. The condition varies. And in most of the real-time machine, you have a given set of thermal condition, which generally works, but then that you can always play with, in particular with the annealing temperature. But in our lab, we are trying to keep constant 60 degree, which is very much standard for the annealing temperature of the primer in the real-time PCR. Make sure that in the software you label the sample correctly. Like, for example, unknown sample with what gene it is, negative control, positive control, and then the mentioning of the gene and the sample number is very much important. And before to start the reaction, you must look that whether you have set it correctly or not. That is the initial denaturation, for example, a hot start, and then followed by 39 cycles of 95 for 30 seconds, let's say, and 60 degree for 30 seconds. And then the last stage is in this image. This last step is nothing but the melt curve analysis. For the melt curve analysis, from 60 degree to 95 degree, or 95 to 65 degree, it will go every 0.5 degree Celsius decrease, right? And that helps in getting the melt curve analysis. And what this melt curve is, we will anyway see when we do the analysis part, right? Troubleshoots. So there could be a problem like sometime you may not get amplification, the amplification plot may look abnormal. There could be primer dimer, non-specific binding, contamination and negative control, no amplification and positive control. All these problems can come, and each of them can be can be solved if you know the basics behind it very well. For example, let's say if RNA itself is not coming, then it could be that whether your reagents may not be working, whether you are not working properly in the laboratory to get the RNA. Once you get the RNA, whether you are doing a good, correctly the DNase treatment, that is again very much important. Even if you do the DNase treatment properly, you have to run the gel and check that whether the DNA is still not there. And then everything what you are using it for the cDNA conversion is RNA. That is again very important. You made the cDNA, but let's say the cDNA itself is not synthesized. So before to go with the complete setup of experiment, you always should test the cDNA. Now, cDNA is working, but sometimes it is possible that the primer what you are using is contaminated or not working, as a result of which, or the primer which you are using may be not specific or having less specificity to your target site. It may lead to dimer, it may lead to non-specific binding, right? If you are not handling your tips, pipettes, your plates, tubes, everything properly, then there is a possibility that the negative control gets contaminated either with the contaminated reagents or by mistakenly you have put the DNA in there. So those things you have to check. In the positive control, if there is no amplification, it could be that the positive control template is degraded, or you have forgotten to add any of the primer or the template itself, right? Cyber green. So everything, all those things needs to be looked very carefully for the troubleshooting. To learn more about troubleshooting, I would suggest that go into this Sigma Aldrich reference, which I am sharing here in this particular slide, and then look for the troubleshooting, different types of troubleshooting which generally happens in the PCR. So with that, I believe that I have covered most of the aspect of the wet lab work, right? Starting from sample preservation to the qPCR setup, and then in the next videos or in following video, you will be able to see how do we what do we do with the output of the thermocycler or the real-time machine, and how do we do all the analysis part, right? So that we will see in the future videos. I am also giving you here the reference for three different things which are very much necessary in the laboratory. And if you buy directly from the leading companies, these are very expensive chemicals. For example, decontaminating solutions, RNA ladder, and the triazole. And all three of them works well. So we have to make in the lab, and yeah, so we have to make in the lab, and we'll try that. Okay, so I guess that's it for this particular session, and remaining things we will see later. Thank you.